but
Yes,
just
sir.
give me a couple of seconds, please.
Okay.
We are live and recording. Thank you very much.
Hello, everybody. It's my great pleasure today to talk to you about the gut microbiome, and in particular to focus on benefits of the microbiome beyond the gut. This is the basic layout of my talk. I'll begin with some discussion of the basics of the microbiome and will address what is normal or what we know about what is normal. Now, there are multiple microbiome impacts beyond the gut, so I've had to be selective, and I'll focus on metabolic disorders, the liver, the brain, and cancer. So let's start with the basics. So why is there all this excitement about the microbiome? Well, basically, this is all technological. And basically, what's happened over the last few decades is that we moved from an era, like when we were medical students, when the analysis of bacteria was based on the plating and culture and analysis of bacteria like this. Now, thanks to the advent of high-throughput sequencing and related technologies, we can now sequence the entire genome of bacteria and other microbes and define their function as well. So it really is a technological revolution. Now, one of the most commonly used techniques is based on the amplification of these 16S RNA genes here, shown in the center of this figure. And this is a highly conserved area in all bacteria, but differs between bacteria. So by using this, you really provide a fingerprint of different bacteria in the gastrointestinal tract or indeed anywhere. And then from then you can go on and do various other techniques to define the genetics or the metabolomics of the sample in more detail. So let's talk about some basic definitions. Now, here, unfortunately, there is some confusion. Strictly speaking, the term microbiome refers to the entire habitat, including the microorganisms, their genomes, and the surrounding environmental conditions. But in fact, in the literature, the term microbiome is usually used to refer to the genomes of microorganisms. And there are other terms. Microbiota should be used exclusively to refer just to the bacteria in a defined environment. The metagenome to the collection of genomes and genes from members of the microbiota, and the metabolome to the metabolic products of the microbiome. But unfortunately, the terms microbiome and microbiota are often used interchangeably, even in the microbiological literature. So don't be surprised, don't be annoyed if I make that mistake also in this presentation. Microbiome and metagenome are often used to refer to genomes alone, whereas strictly speaking, only metagenome should be used for this term. Now, I'm not a microbiologist, but I have to read this literature. So here's how I interpret the various methods that are used in a very simplistic way. The most commonly used technique still is high-throughput sequencing, and that basically tells you what's there, albeit not at a very highly defined level. It gets to certain levels, but beyond that, it's not very accurate. Metagenomics, which defines the genetic material of all bacteria, will tell you what they could do by predicting function from genes. Metabolomics and metatranscriptomics will actually tell you what they produce. As we move forward in this area, we see more and more studies which combine metagenomics and metabolomics or metatranscriptomics. So what is normal? Well, I want to share with you a few studies that have looked in detail at large populations of normal individuals to try and define what the variation between individuals is. And this is a study at genus level from Europe, which defined a number of variations at genus level. Remember, this is at a very high level of organization between individuals based on the dominance of Bacteroides, Prevotella, or Ruminococcus, as you can see here. And what they suspect is that this level of organization is probably largely driven by diet. What most studies agree on is that in normal human beings, the Microbiome, or microbiota, I should say, at phylum level is dominated by two phyla, Bacteroidetes and Firmicutes, as you can see here, which much smaller representation from others. And this balance between Bacteroidetes and Firmicutes has actually been used to analyze changes in the microbiome in disease states. Now, this is a very interesting and important study, which is very informative. And I'll tell you a little bit about the background. So this is a study which was performed in 3 different areas: in cities in the US, in the Amazon basin in Venezuela, and in rural Africa in Malawi. And what they looked at is the variability or variations in the diversity of the microbiome over the lifespan of these individuals and then between these 3 groups. So over here we've got it on the y-axis, we've got the number of OTUs. What that basically means is the number of different bacterial species or strains, so the greater diversity. And as you can see here, for all these populations, as we move from birth, time 0, to old age, you see a dramatic rise in the number of bacterial species over the first couple of years of life. And this is actually shown in greater detail in the little figure below it here. And here you can see actually an exponential increase in the number of bacterial species from age 0 to 3 years of life. So that's the first important observation, is that in normal individuals, you see a dramatic development of the microbiome in the early years of life. What you can see then is over the rest of life, it remains fairly stable. But there are differences, as shown in the columns down here on the part C. And what you can see here is that the number of individual bacterial species was much greater in the Amerindians and the Malawians than in the urban US dwellers, whose diversity was much lower. And in general, diversity is a good thing. The more diversity in your microbiome, the healthier you are. And again, this differences between the USA and the other 2 groups was thought to be largely diet-driven, with the US urban dwellers eating a highly processed diet, whereas the Amerindians and the Malawians were probably eating a much more plant-based diet. Now, there are other issues which will influence the results of any study on the microbiome. It depends where the sample is taken. Is it fresh or frozen? What technique is used, and how good are your informatics? Because these studies generate enormous data sets which require highly specialized informatics to analyze them properly. Now, how about location? Well, as we all learned in medical school, there are dramatic changes in the abundance and diversity of bacteria as you move through the gastrointestinal tract, from the oral cavity through the esophagus, stomach, and small intestine, and then most dramatically as you enter the colon. And of course, these changes are influenced by the luminal contents, by the acidity of the environment, its bile content, the content of digestive enzymes, how quickly or slowly material moves through this area, its oxygen concentration, and the availability of nutrients. For example, the colon is a highly anaerobic area and favors anaerobic bacteria. And this shows you figuratively the changes in the bacterial concentrations as you move from the duodenum and jejunum into the terminal ileum with a big increase, and then the greatest increase as you move into the colon, as you can see here. Now, the other issue which is very important when you're sampling from the gastrointestinal tract is, are you sampling from the lumen or are you sampling from the mucosal area? So obviously the bacterial numbers, not obviously, but it's been shown in many studies that the bacterial population out here towards the lumen is quite different from that in the juxtamucosal area. Now, what is the role of the microbiome in health? There are several important functions which have been recognized, and no doubt more will also be discovered. So I've summarized them on this slide. First of all, your normal microbiome prevents ingress of pathogens. The presence of an intact normal microbiota is key to the development and maintenance of the immune system, and in particular the generation of tolerance. As we move forward in this area, we recognize several metabolic roles for the microbiome: the production of short-chain fatty acids, the metabolism of bile acids, the production of other metabolites, including neuroactive compounds, and importantly, participation in the metabolism of a variety of drugs. We also recognize that an intact microbiota is important for the development and integrity of the gut barrier, the enteric neuromuscular apparatus, and even the central nervous system. Now, what influences the microbiome? As already mentioned, age is critically important. The first few years of life witness the dramatic development of the microbiome. Birth mode, cesarean section versus vaginal delivery, infant feeding, breast versus formula, diet, antibiotics, and undoubtedly other medications, as well as geographic location and disease. And I just want to emphasize disease for a minute because this is very important. Just because you see a change in the microbiota in a given disease state doesn't mean it's causing the disease. It actually could be a consequence of the disease. Now, when we go back and think about these factors, it is critical to important to understand that several of these factors have a very crucial impact in early life at a time when the microbiome is very vulnerable, because that's at the time that it's actually developing. And diet indeed is a major factor. In the long term, for sure, the diversity of the diet, its fiber content, its protein content are critical. For example, the fat content, as it increases, leads to a reduction in lactobacilli and bifidobacteria, which are regarded as critical commensal bacteria. But even in the short term, diet can change the microbiome if the change is sufficiently drastic. For example, going from a meat-eating diet to a vegetarian diet or a vegan diet. Just one word about antibiotic exposure. This is a very important recent study which followed a very large number of patients from before, or children, I should say, from before birth to the age of 4.5 years. By age 4, 95% of them had been prescribed an antibiotic and 9% are obese. And they found a direct correlation between the number of antibiotic exposures and BMI, particularly if they were exposed under the age of 1. If they had more than 9 prescriptions for antibiotics, the odds ratio for obesity was 2.41. So this illustrates what I said earlier, namely the vulnerability of the microbiome at that early stage in life. And there are several other studies which have suggested that impacts or insults to the microbiome early in life have been associated with development of inflammatory bowel disease and irritable bowel syndrome later in life. Now, how about the microbiome and obesity? Well, interest in this area actually goes back quite a few years now with these key observations in animal models. First of all, it was noted that the microbiota of this genetically obese mouse called the OBOB mouse was different to lean mice. Secondly, it was noted that an obesity-inducing diet decreases the diversity of the microbiota, and then the critical observation that the obese microbiome was highly efficient at extracting energy. So here you can see the gut microbiota having a critical level in a critical role in several factors which are involved in the development of obesity: the gut barrier, adipose tissue, liver, even at brain level and even at the level of muscle. What's been very interesting has been the number of studies which have demonstrated that these characteristics, these impacts in terms of obesity, are transmissible, therefore can be transmitted from the microbiota. This is showing here twins, one of whom is obese, one of whom is lean. If you transplant microbiota into mice, the microbiome from the obese twin, the animal develops some adiposity. The opposite happens if you transplant from the lean co-twin. So how does the microbiome actually contribute to obesity? Well, there are several mechanisms have been identified. I already mentioned the extraction of calories, and basically bacterial fermentation producing short-chain fatty acids and monosaccharides activate protein YY, which retards transit, and activates these other signaling molecules, which I'll illustrate on the next slide. It also needs the suppression of fasting-induced adipose factor and activation of AMP-activated protein kinase. And these are illustrated here. You can see the degradation of complex carbohydrates leads to production of short-chain fatty acids, which activates PYY, which will retard transit and therefore promote energy harvest from the diet. The production of monosaccharides activates this carbohydrate responsive element binding protein shown here, which in turn will lead to increased lipogenesis and increased gluconeogenesis. It leads to suppression of fasting-induced adipose factor, which leads to increased fatty acid uptake into adipose tissue and decreased beta oxidation, all of which will contribute to obesity. Other mechanisms including increased intestinal permeability. A high-fat diet impairs permeability, whereas in contrast, the commensal organism Akkermansia eosinophilia protects against this. Immune activation has been described in obesity. In fact, some would refer to it as a pro-inflammatory condition, and has been shown that a high-fat diet alters microbiota and increases lipopolysaccharide levels, leading to weight gain and insulin resistance, and of course leads to metabolic endotoxemia. Now, this summarizes some of these factors in this slide, and you can see here that with obesity, you get impaired intestinal barrier, bacteria or bacterial product translocation, shown here, activation of toll-like receptors, shown here, ultimately leading to the release of pro-inflammatory cytokines such as TNF-alpha, interleukin-6, and leptin, leading to the activation of hormonal axes and contributing to obesity. Other things that are going on here, again, dramatic systemic effects of microbiota include the impact on the endocannabinoid system, modulating intestinal endocannabinoid tone, and of course the role in bile acid metabolism, and with the microbiota metabolizing bile acids via bile salt hydrolase. Bile acids are bacteriostatic, therefore inhibiting the microbiome, and bile acids also act as signaling molecules involved in appetite regulation. So what I'm saying in all of this is that there are multiple systemic effects of the microbiome which contribute to obesity and fat accumulation. Now, what about studies in man? And this is a large study which looked at the diversity of the microbiome as illustrated by the number of genes between control subjects and those who are obese. And as you can see, there are significantly fewer genes in patients who are obese compared to controls. And this is shown here in terms of accurate gene count and alpha diversity. But this may be reversible, and this is illustrated in this study, which looked at what happens with patients undergoing bariatric surgery. So here are control individuals with the probability of obese, which is obviously very low, then the obesity here, then followed by bariatric surgery, and then the reduction in weight over 3 months following surgery. And this is in association with this, we see significant changes in the bacterial species with a return towards a more normal microbiome. Now, with regard to the microbiome in the liver, we've known about the role for the microbiome in the liver for many years, in particular in relation to the enterohepatic circulation. Because normally, as we know, most bile salts are reabsorbed in the terminal ileum using this intestinal bile acid transporter and recirculated to the liver. A small amount are lost, and those that are lost are then deconjugated by bacteria in the colon to produce secondary bile acids, which actually promote diarrhea through increasing secretion and also by increasing motility. And this has been referred to as the Microbiome-Gut-Liver Axis, but now this has been extended to involve a lot more than just bile acids. So for example, here, and not only when our bile acids being reabsorbed, but also this is stimulating this molecule FXR and FG19, which in turn has direct effects on bile acid synthesis in the liver, as shown here. At the same time, from the gut, lipopolysaccharide, endotoxins, and bacteria gain access to the portal circulation, leading to the activation of toll receptors and Kupffer cells in the liver. This is shown in more detail here, where we've got in the gut lumen, if you've got a disturbed and altered microbiota, perhaps in association with the change in gut barrier function, can lead to an immune response first in the gut, and secondly, the translocation of bacteria, endotoxins, and cytokines to the liver, where they can activate an inflammatory cascade through binding to toll receptor 4 and leading to inflammation and other changes in the liver, and potentially to chronic liver disease. One of the areas that this has been worked out in most detail is non-alcoholic fatty liver disease. Many studies have shown that small intestinal bacterial overgrowth is common in patients with non-alcoholic fatty liver disease, as shown here. Others have shown there are actual qualitative changes in the gut microbiota in patients with obesity, which of course is closely related to non-alcoholic fatty liver disease. And they've also shown that gut bacterial metabolism of alcohol, leading to the production of alcohol, acetaldehyde, trimethylamine, and trimethylamine oxidase also occurs and have potential effects on the liver. When you have all of this in association with impaired gut barrier function, It results in translocation of bacteria or bacterial products such as lipopolysaccharide, as shown here. Activation of inflammatory cytokines, as shown here, which also enter the portal circulation, leading to an immune response to the microbiota, with toll receptors playing an important role. And all of this cytokine release, of course, has the potential to produce an inflammatory response in the liver, particularly TNF-alpha. Now, with regard to NAFLD and the gut microbiota, there's important evidence to illustrate that there is indeed an important role for the gut microbiota in non-alcoholic fatty liver disease. So here's a nice study where they gave mice a high-fat diet, and they found that some responded to this by becoming obese and some did not. Then they transplanted microbiomes from non-responders and responders into normal recipients, germ-free mice. And what they found was when you did this, that in those who got the microbiome from the responder group, they developed steatosis, as shown here, and is measured here in the steatosis score. Just as I showed earlier for obesity, it's actually also been shown that you can transmit a tendency or a predisposition to non-alcoholic fatty liver disease through the microbiome. So here you've taken a group of normal weight women and obese women, and then you've studied them, you've sampled the microbiome from their children and transferred it into mice. And what you can find then, when you do this, is that you get significant changes in short-chain fatty acid production, significant changes in gut permeability, as shown here, with increased permeability in the obese mice, and this is associated with more bacteria translocating to the liver and more fatty liver, as shown here. You can see here the changes in the liver in association with this. Not only does this happen, but if you add a Western diet to these animals, now you get a dramatic increase in fat content in the liver, showing the convergence of microbiome and diet in terms of producing fatty liver disease. Now, this may surprise you that I even discussed this topic, but there's a lot of interest now in the potential impact of the microbiome on the brain. First of all, before we get to the big brain, let's say a word about the little brain, in other words, the enteric nervous system. And there's a lot of evidence to show that the microbiota plays an important role in the development and function of the enteric nervous system, probably through its interaction with the immune system in the submucosa and in the production of various cytokines. This is the concept of the Microbiota-Gut-Brain Axis. So here you've got the gut, and if you've got a healthy gut, you've got a healthy central nervous system, normal behavior, cognition, emotion, nociception, healthy levels of inflammatory cells and mediators, and a normal gut microbiota. On the other hand, if your microbiota is disturbed, it can lead to abnormal CNS function and abnormal gut function, and all of these bad changes. Well, one thing that's very important to recognize about the gut-brain axis is that it's bidirectional. There's a lot of research to show that the microbiome can influence brain and behavior through activation of neuroafferent circuits, through activation of mucosal immune responses, and through the production of metabolites that directly influence the CNS. But equally important to recognize that the brain can influence the microbiota through inducing changes in gut physiology, epithelial function, mucin production, enteroendocrine cell function and motility, and the release of neurotransmitters. Now, I've taken one example here. I know the irritable bowel syndrome is a gut disorder, but it does actually exemplify the many ways that the microbiome can influence the body in a general sense. So obviously, the microbiome can lead to immune activation. It can lead to changes in gastrointestinal motility and sensation. And as I've said over and over again, it can lead to changes in intestinal barrier function. But it can also lead to alteration in the gut-brain axis, and in this way influence symptoms in irritable bowel syndrome. But this is very complicated because there are many other factors that can influence this: host genetics, stress, diet, antibiotics, and early childhood experience. Now, this is a very interesting study which illustrates how a very subtle change in the microbiome, in this instance giving just a single probiotic organism, can influence brain function and actual symptoms. So this was looking at patients with irritable bowel syndrome who also had comorbid anxiety and depression. And what they showed basically was that by giving the probiotic, you could significantly decrease This is the so-called hospital anxiety depression scale, the score for depression at 6 weeks and at 10 weeks in these particular patients. So you saw a significant clinical effect. It also led to improvement in quality of life, but actually had no impact on IBS symptoms. It didn't significantly change the fecal microbiota or serum markers of inflammation or levels of neurotrophins or neurotransmitters. So at least from that point of view, the mechanism here was unclear, but it was associated with some metabolomic changes, which is an important point, important general point, because the expectation that a single probiotic is going to change the composition of the microbiota is just not going to happen. So it's much more likely that there are going to be subtle changes. And very interestingly, they went on and did brain imaging, and they showed that this probiotic, as I've shown you, could actually influence brain activity in different parts of the brain. And it's one of the few demonstrations that I know of microbiome-gut-brain axis in a clinical context, as opposed to an animal model where there's been a lot of data. There was no impact on IBS symptoms, but it was underpowered to do that. And the effect was not related to changes in the composition of the microbiome or the immune response, but more likely was related to a change in the function of the microbiome. Now, the next study is a bit more complicated, but looks at the stress response. And here we're looking actually at normal individuals, and they're asked to do a task which is very stressful. It's called the cyberball game. And this was performed under two conditions. First of all, when they were given a placebo, and secondly, when they're given a probiotic, B.longum. And what they showed basically was that at baseline, the B.longum actually was associated with some change in neural activity. But more importantly, when the same was done during stress, this is actually during the stress here, I apologize. You can see that there are significant differences in the response, in the brain response to stress, in the group who got the placebo compared to the probiotic. And you can see here, in fact, there was a nice correlation both at rest and, more importantly, in response to stress between the neural activity and the symptomatic response in those who were given the Bifidobacterium-longum. Complicated study, but basically what it's showing is that a single intervention here, this time with a probiotic, can influence the stress response in normal individuals and result in a reduced stress response in the brain. There have been many studies looking at the microbiome and neurological disease. Parkinson's, Alzheimer's, Huntington's, multiple sclerosis, amyotrophic lateral sclerosis, autism, depression, anxiety, and schizophrenia. Many of these studies suffer from the problem that I mentioned earlier, namely, they cannot differentiate between what is an association and what is actually causation. But I want to mention a few interesting things. This is a very interesting study which looked at a mouse model of Parkinson's disease. And this is produced by overexpressing alpha-synuclein, which of course has been associated with Parkinson's disease in man. And what they showed was that antibiotic treatment ameliorates and restores the microbiota in these animals, and they get rid of the microbiota that seem to aggravate motor deficits. What they also showed is that if you took microbiota from patients with Parkinson's disease and gave it to these mice, as shown over here, it actually enhanced motor dysfunction, suggesting that the microbiota has a role in Parkinson's disease and that this can actually be transmitted. I want to finish with a few words about the microbiome and cancer. Now, the idea that a microbe could be involved in cancer is not new, of course. We know about Helicobacter pylori and gastric cancer and multiple lymphoma, and multiple viruses, human papillomavirus, Epstein-Barr virus, hepatitis B and hepatitis C virus, human herpes virus, HIV, and human T-cell leukemia virus have all been associated with a variety of cancers. Now, how does the microbiome actually influence cancer? Well, there are multiple mechanisms that might be involved here. They vary from genomic integration, where you get integration of microbial DNA into the host genome. And the example here is human papillomavirus virus, which inactivates tumor suppressor genes. Another mechanism is genotoxicity, whereby the microbiome induces structural DNA damage. And this has been shown with cytoleucine distending toxin from Campylobacter jejuni and Escherichia coli and Colibacter enterobacteriaceae. These are carcinogenic in animal models. A very common way, of course, is inflammation. We know that inflammation is associated with a lot of cancers. An example here is Fusobacterium nucleatum associated with colon cancer. It can suppress immunity like HIV or can result in the production of various metabolites such as aromatic amines, sulfides, and acetaldehyde, which can be associated with genotoxicity. The one you all know, of course, is Helicobacter pylori, and there are very complex interactions here between the bacterium, various bacterial characteristics, and the host immune response, which result in mitogenic and pro-inflammatory transcription and can lead to the development of cancer. Putting it simply, Helicobacter pylori is a very nice example of the interaction between the bacterium, its environment, the host response, which is probably genetically determined, and outcome, i.e. whether an individual who has Helicobacter develop cancer, a duodenal ulcer, or gastritis. Now, this nice slide here illustrates the many associations that have been claimed between various microbes and gastrointestinal cancer, from oral cancer through hepatocellular cancer, gallbladder cancer, colon cancer, gastric cancer, esophageal cancer, etc. So multiple proposals, not all of them supported by sufficient data, but certainly very intriguing data. Now, when we come to talk about the microbiome and cancer, indeed the microbiome in any area, we've got to think about what are its possibilities for the future. Well, one of the areas that's really exciting people is the possibility of using the microbiome to diagnose, to differentiate between healthy individuals and those with disease, to identify a disease subgroup, for example, those who have a good prognosis versus a poor prognosis, or those who may respond to a particular therapy or not respond to a particular therapy. The other area, which of course has been studied for some time now, is the potential role of microbiota or microbiota-derived products in therapeutics. And this can occur in two ways, either by modulating the microbiota or by mining the microbiota for actual new therapies, such as probiotics. Now, I want to mention a few very intriguing examples here, which really fascinate me. This is a study where this group from Ireland actually looked at the oral and the colonic microbiome in patients, not just with colorectal cancer, but also with colon polyps. And what they found is just using the oral microbiome, this is not the colonic microbiome, just the oral microbiome, they found that they could clearly differentiate between cancer and controls and those with polyps. And this is showing here the difference in relative abundance between these groups. Very interesting. They then went on to look at this in more detail and actually looked at the diagnostic potential of the microbiome in predicting those with colorectal cancer or an adenoma. And they did this by combining the oral microbiome results and the stool microbiome results. And so, for example, here you can see here the results from the oral, the stool microbiome, and then when you combine them together, and these receiver operating curves, the closer you get to here, of course, the more accurate it is. You're getting very good sensitivity and specificity. Sensitivity of 53%, and more important, specificity of 96%. And this was based on 16 oral microbiota OTUs. And even for adenoma, you found the same thing. This is you got the oral was probably not as good in terms of adenoma detection, but when you combine it with fecal, you see again here very high rates of sensitivity and specificity. Now, obviously, this study needs to be repeated, but this is very exciting and suggests that you can actually use the microbiome to detect cancer and, more importantly, detect a precancerous stage, namely the adenoma. Now, this study absolutely fascinated me. So this is looking at a very new and very exciting form of therapy in cancer, namely immune checkpoint inhibitors. This looked at 3 different checkpoint inhibitors: ipilimumab, nivolumab, and pembrolizumab. Apologies if I'm mispronouncing these, but they really are tongue twisters. And what they looked at were 39 melanoma patients, 23 of whom actually responded to 1 or more of these checkpoint inhibitors, and 16 who failed and had progressive disease. And then they compared their gut microbiomes and metabolomes between the 2 of them. And what they found was that the microbiome was highly predictive of the response. Whether, regardless of which of these agents you looked at, the responders were enriched for certain bacterial species, such as Bacteroides caxae in this example, Faecalibacterium prausnitzkii, Bacteroides theata or iotomicron in this one, or Holdemana filiformis for this particular one, or in this particular checkpoint inhibitors, Doria formicogenerans. So basically, the baseline microbiome before therapy could predict those who responders. Metabolomics were also predictive in that those who responded had high levels of anacardic acid in their microbiome. There have been a number of other studies which have looked at the microbiome as a predictor of response to a high-fat diet, as a response to a high-fiber diet, as influencing both the prevalence of side effects and response to metformin. And I think we're going to see many of these interactions between the microbiome and response to various therapies, which could really revolutionize what's referred to often as personalized medicine. So I hope you've been able to bear with me as I've really taken you through a fairly broad sweep of various aspects of the microbiome. So the fundamentals of the microbiome have been known for some time. What has changed are dramatic and ever-evolving developments in technology and informatics, which are really driving the current explosion in knowledge and understanding. The metabolic contributions to the microbiota are considerable and include bile salt metabolism, short-chain fatty acid production, and even the production of neuroactive compounds. The gut-liver axis is bidirectional. Bile acids are important regulators of several homeostatic functions. Changes in the microbiota, altered intestinal permeability, and gut and liver responses conspire to produce or induce obesity and related disorders. Considerable animal and some human data suggest a role for gut microbiome in neurological disease and cancer, but as I've emphasized repeatedly, be wary of associations. They do not prove causation. There are many confounders which hamper interpretation of microbiome studies, including diet and the disease itself. Microbiota analysis holds promise in diagnostic, and tantalizing hints of this promise continue to emerge. What I've not had time to talk about is the importance of microbiota modulation and mining the microbiome, because both of these offer unlimited promise, but at the moment high quality trials are relatively few. And with that, I'd like to thank you for your attention.
Yes, sir. Thank you so much for such a nice presentation. And from the core of our heart, we would like to say thank you very much, once again for, delivering the talk. And definitely on the 21st, we are going to be meeting again for, initially there is going to be this pre-recorded session and followed by the question and answers. So for question answers, you need to be available, for interaction with the audience.
Okay.
Yeah, thank you so much.
Okay.
Good day, sir.